1,137,645. Okay, I know, it's a massive number. But what if I told you that this figure doesn't represent, stars in the sky or people in a city? No, this is actually the exact number of high-quality DNA sequences that researchers just pulled from the microscopic guts of only 17 tiny ground beetles. Welcome to this explainer.
So, can the microscopic gut of a beetle actually tell us how healthy our planet is? It's a pretty mind-blowing question to pose, isn't it? We're setting up a bit of an eco-detective story today. And as we decode the DNA data pulled from these specific insects, you're going to see exactly how a completely hidden, invisible ecosystem basically shapes the physical world we rely on every single day.
The microscopic dashboard, secrets of the Poecilus beetle. What researchers found hidden in that incredible mountain of DNA data? Well, it might literally change how we look at agriculture, soil health, and just the overall interconnectedness of nature forever.
Section 1. Meet the tiny ecosensor. Okay, let's dive into this by introducing our unlikely hero. Because before we can really understand the microscopic data, we've got to understand the macroorganism we're dealing with. So, here's the Poecilus beetle's resume. This isn't just your average garden bug. It's an absolutely vital natural pest controller. The researchers specifically collected these guys from tomato fields, where they're actively patrolling the rows and munching on agricultural pests. Think slugs, caterpillars, aphids. And because they're such voracious predators, having a healthy population of these beetles naturally reduces a farmer's reliance on chemical pesticides. They are a totally crucial ally for agriculture.
Because of this specific role, scientists look at these beetles as a bioindicator. Now, think of a bioindicator as a living, crawling environmental sensor. Just like the dashboard of your car has a check engine light to tell you what's happening under the hood, right? Well, the presence, the health, and crucially the microscopic makeup of this beetle acts as a biological dashboard that we can actually learn to read to understand the overall health of the soil.
Section 2. Inside the microbial engine. So, let's take a little journey from the macro environment of that tomato field right down into the micro-engine room of the beetle's gut. This gut isn't just a simple stomach digesting food. Actually, scratch that, it's more like a highly complex factory. The DNA sequencing revealed a core microbiome made up of absolute powerhouse bacteria. You've got Gilliamella, which brings the superpower of carbohydrate metabolism and breaking down plant compounds. Then there's Weissella, which is absolutely essential for nitrogen cycling and fatty acid synthesis. And Enterobacter, providing powerful resistance to toxic compounds. I mean, these microbes are actively synthesizing amino acids, producing vitamins, and neutralizing chemicals for the beetle.
Now, what's really interesting here is how this scatter plot visualizes the stability of this engine room. The data from the study maps out the prevalence and abundance of the gut bacteria. See those red dots hovering way up in the top right corner? Those are the absolute VIPs, the core taxa like Gilliamella and Weissella. The data shows they're highly conserved, which basically means they show up consistently across the beetles to keep them healthy and functioning, regardless of minor environmental differences.
And this bubble plot brilliantly illustrates the chemical superpowers I mentioned just a second ago. It maps out the predicted functional scores of the gut microbiome for each individual beetle. Basically, the larger and brighter the bubble, the higher the predicted activity for things like amino acid synthesis and protein degradation. We aren't just identifying the bacteria here. We're literally mapping out the metabolic heavy lifting they're doing to keep the beetle thriving in a dynamic agricultural landscape.
Section 3. The surprising pathogen plot twist.
You know, in every good detective story, the evidence eventually points to something no one was even looking for. As the researchers sifted through those million-plus DNA sequences, they found something incredibly surprising. They found a glitch in the matrix, a complete anomaly that proves the ecosystem holds completely unexpected, invisible secrets.
Look closely at this pathogen presence chart. That top row shows the detection of a pathogen called Nosema ceranae. Do you see how it's present in every single one of the 17 beetles? Guys, this is a jaw-dropping finding. Nosema ceranae is a well-known pathogen of honeybees. This marks the very first time it has ever been detected in a Carnobacterium ground beetle. They also found Ceraradia, another opportunistic pathogen, in 6 of the individuals.
So we have to ask the obvious question. What on earth is a famous honeybee pathogen doing inside a ground beetle? It sparks so much wonder about the hidden interconnectedness of our ecosystems. The researchers suggest that because these beetles are ground-level predators and scavengers, they might actually be acquiring this pathogen by scavenging on deceased, infected honeybees that fall to the soil. It perfectly illustrates how an impact on a pollinator flying way above can directly transfer into the gut of a beetle crawling below.
Section 4. A Tale of Two Farms.
Let's move on and see how this builds as we examine the actual agricultural crime scenes where these beetles were captured. The researchers collected from two distinctly different types of agricultural management. Here's the strict data observed by the researchers. From the organic tomato field, they collected 15 beetles. Through the DNA sequencing, these beetles showed a rich, balanced, and highly diverse gut microbiome. On the flip side, from the conventional tomato field, they collected only 2 beetles. And the microbiome of those 2 beetles showed extremely low diversity, overwhelmingly dominated by a single microbe. Now we're strictly looking at the biological variance here, but the stark contrast in these numbers is just fascinating.
To understand what happened to the beetles in the conventional field, we need to understand the biological concept of dysbiosis. Dysbiosis is a dangerous collapse or imbalance in a gut microbial community. When an organism faces severe environmental stress, like say exposure to certain agrochemicals, the rich diversity of the gut engine basically crashes, and this leaves the host to be dominated by single, often opportunistic microbes.
So the crucial point is perfectly visualized right here. Take a look at this stacked bar chart showing microbial abundance. On the far left, look at sample P1, which came from the conventional field. Notice that massive dark purple spike? That represents a microbe called spiroplasma, and it has taken over a staggering 99.1% of that specific beetle's gut. The diversity is virtually gone. It's a complete monodominance. It's literally a textbook visual of dysbiosis, potentially linked to the environmental stressors of that specific field.
Section 5. The Future of Soil Health. As we zoom our narrative back out from the microscopic gut dominated by spiroplasma to the macro world of the tomato field, we start to see the global scale of our agricultural future. The researchers ultimately concluded this, saying, these insights support the use of Poecilus as a bioindicator for soil ecosystem health and highlight the potential of microbiome-based metrics in agroecological monitoring. That is their ultimate breakthrough. This tiny insect isn't just a bug. It's a living, crawling mirror of the soil that grows the very food that ends up on our plates. By reading its gut, we literally read the health of the farm.
Which leaves us with one final incredible thought to end our explainer today. If a simple beetle's microscopic gut holds the complex, interconnected secrets of our soil, our pollinators, and our farming practices, well, what else is hiding right beneath our feet, just waiting for us to sequence it? Thank you so much for exploring this with me today. Keep wondering and keep looking closer.